So, hello, good day. I am Mark Elis Artayaw from BSN 1A. I'm going to discuss to you about the physiology of muscle contraction. So, for our core concept, we have the muscle contraction, and the sliding filament theory. So, before we proceed to the main topic, let us define first what is muscle. So, it is a tissue composed of cells or fibers and the contraction of which produce movement in the body. For our muscles in the body, we have three different types of muscle, where we have the skeletal muscle, smooth muscle, and the cardiac muscle. So the difference between those three is skeletal muscles are voluntary muscles, unlike the cardiac and smooth muscles who are involuntary muscles. So for muscle contraction, it occurs when tension-generating regions within the muscle cells are activated. So, muscular contraction does not always imply muscle shortening in physiology, since muscle tension can be generated without changes in muscle length. Some example activities that shows how muscle contraction occur. As you can see in the PowerPoint, we have lifting, running, stretching, and push-up. All of these activity make the muscle contracts. You can see in the screen, it is the contractile unit of the muscle or a myofibril that can be found in skeletal muscle cell, where it has a thin and thick filament. The thin filament is called actin filament, which is the color blue, and the thick one is called myosin filament, which is the color red. You can also see an eye band in the middle of it is what we call the saline or the satis. Eye band only contains thin actin filaments, while on the next one we can see the A band, which contain the H zone or what we call the bear zone. In the middle of it, the line shows as the M line. Unlike the I band, A band contains both thin and thick myosin filaments, and the distance between the two Z line or Z disc is what we call the sarcomere. So, on the screen, it shows how muscle contracts. In figure A, muscle contraction does not occur, while in the figure B, we see that the muscle contraction occurs. We will see that the thin actin and thick myosin filament slide past each other. By the way, here the red one with heads is what we call the thick myosin filaments, while the blue one are the thin actin filaments. So both of them slide past to each other. That results the disappearance of the H zone or the bear zone. In that case, we can call that the muscle contracts. Sliding filament theory explains how muscle fibers contract. So, myosin filaments uses energy from ATP to walk along the actin filaments with their cross bridges. This pulls the actin filament closer together, and the movement of the actin filaments also pulls the salines closer together. Thus, it results the shortening of the sarcomere. When a nerve impulse reaches to the muscle, it causes the chemical acetylcholine to be released. The protonation occurs when acetylcholine is present, allowing calcium to be released from the sarcoplasmic reticulum. Then, calcium binds to the tropomyosin, changing its structure. The myosin filament heads can now attach to the actin filament, causing a cross bridge. The breakdown of ATP release energy, which enable the myosin to pull actin inwards, contracting the muscle. This occurs among the entire length of every myofibrils in the muscle cells. When an ATP molecule binds to the myosin heads, the myosin detaches to the actin, then the cross bridges will be broken. Myosin heads can reattach to actin binding sites further down the actin filament once the ATP has been broken down, and the process can be repeated. When all of the sarcomeres in the muscle fiber shorten, the fiber contracts. A muscle fiber either contracts fully or it doesn't contract at all. So, the number of fibers that contract determines the strength of the muscular force. Always remember that the more fibers contract at the same time, the greater the force will release.